All right, everyone. So I am very excited today to share a whole bunch of student presentations with you from this October research program. We have 5 great presentations from students across California who all have these really neat science projects that they've been working hard over the last month or so to share. But before we get into those, I just wanted to share a couple things with all of you about kind of how this will go, and a little bit more about what the work that all of these students have done so far. And so each one of these students has a kind of 6 to 7-ish, maybe a little bit longer presentation that they'll share with you. And then at the end, we'll have a chance for you as an audience to ask these students questions about their research. As we go along through the talk, you can comment any questions that you have, and we'll have a Q&A session at the very end. And so feel free to comment your questions whenever, but we might not get to them until the very end. I think we'll have time for a handful of questions for each presentation before we get going. And so for those of you just joining, I'm Spencer Houston. I'm the program director for Headwaters Science Institute. And I've had the pleasure for working with these 6 students that you'll see today over the past month as they design their own research projects. And one of the really special things that you'll see as we go through all of this is that all of these questions are these students' own. We have a whole bunch of questions, and these students all just pick something that they were really interested in doing. And so that's why we have questions on water quality to earthworms to wildfire and soils. And so this is kind of a neat range of different student interests. And so before we get into the presentations, I just wanted to share that for any of you who are interested in doing something like this, we have another similar type of program that's a little bit longer that's running this spring. So from February to April. It's for high school students, and what we're really going to do is take this similar type of project and help students collect their own data and write a scientific manuscript from that. And so they'll kind of collect all their own data, do these great experiments just like these students, but then they'll also, in the extra couple months that we have for this program, write a manuscript to submit to a scientific journal. And so these students will kind of get not only a really cool research project, but also they'll ideally publish a paper from what they're doing. And so with that, I just wanted to introduce the students that we have going first today. And then we have Caroline and Grace. We have a little bit of a double presentation. These 2 teamed up to create a really awesome project that I'm excited to share. Caroline and Grace, I don't know if you can share your screen in the background, and I'll pull this up for you too. And I'll pull myself out as Caroline and Grace share their presentation. You are welcome to comment questions and they'll answer them at the end. So with that, I'm going to pull up their presentation and introduce Caroline and Grace. If you both want to unmute yourself and get going, I think we're pretty much ready to go. Any questions from the 2 of you before you get going? Great, I'll take myself out of here, and I enjoy hearing your presentation.
Hello, everyone. My name is Grace, and this is Caroline, and we are excited to share with you our comparative study between soil quality in Bayview, Hunters Point, and Crissy Fields.
The Marina Green. 74 acres of grass with a view of the Golden Gate Bridge. The average home value in the marina is $2,100,000 with a racial demographic of predominantly white. It is now regulated by SF Parks and Rec. The Marina Green, also known as Crissy Field, first belonged to the native Ohlone. Then when the U.S. arrived in 1846, it became an airfield and a military industrial area. The airfield was closed in 1974, and the marina was later restored in 1994. Even as Grace and I were collecting data around the wildlife preserves, we could observe sandpipers and great egrets among dogs and children playing on the beach.
Here are the 2 locations our study takes place. Bayview Hunters Point is located in southeast San Francisco. It is geographically enclosed by freeways and holds the Hunters Point Naval Shipyard. The neighborhood has historically been an epicenter for industrial and military activity. The Navy uses the shipyard to decontaminate radioactive ships and military equipment, and has studied the effects of radiation on animals and materials. They gave animals lethal doses of radiation and combined, stored, and burned radioactive fuel. The byproducts of these activities still remain in the soil, causing the EPA to declare the shipyard a Superfund site. A Superfund site is land that has been contaminated by waste that poses a risk to human health or the environment and is eligible for federal funding to clean it up. During and after World War II, the shipyard provided well-paying jobs drawing a large number of African American residents in the neighborhood, resulting in Bayview Hunters Point becoming one of the only predominantly African American neighborhoods in San Francisco. After nuclear testing in the Pacific, as seen by this photo, 79 radioactive ships were brought back to the shipyard, and the employees were responsible for cleaning these ships. These people were exposed to radioactivity. It contaminated their body and their clothes, and as a result, the radioactivity was brought back to their homes and community, exposing exponentially more people. Since the closing of the shipyard, the Navy, who is now responsible for the cleanup of the site, has been prepping the shipyard for a large residential and commercial development space with parks and open space. But things are not going to plan. To oversee the cleanup of the site, the Navy hired Tetra Tech, an environmental engineering firm. Tetra Tech outsourced external workers to gather soil samples who hid the true toxicity of soil taken from the shipyard. The Navy found that nearly half of the soil samples by Tetra Tech were false or manipulated. The EPA questions 97.8% of the data. Tetra Tech will not take responsibility personally. A $27 billion lawsuit was filed against the company recently. Walking around Bayview today, one can see low-income housing, condos developed by Leonard Corp sitting directly above the shipyard, and a desolate shipyard paved over with layers of concrete.
Here are just some of the very many chemicals and heavy metals that may increase one's health risk and make Bayview a toxic and inhabitable environment. These heavy metals and toxic chemicals we tested for are in bold. That's lead, mercury, arsenic, and chromium. Chromium is a mutagen and carcinogen. It concentrates in your blood, bones, and organs and can cause respiratory problems, a lower ability to fight disease, birth defects, infertility, and tumor formation. Humans absorb higher levels of lead when it is breathed in. This is not good considering we found high levels of lead in unstable topsoil in Bayview. Lead is absorbed through and stored in our bones, blood, and tissues. Mercury is a neurotoxin. Long-term exposure to mercury may lead to loss of peripheral vision, as well as many other negative health side effects listed here.
Earthworms. Worms are the most important member of the soil biota. They play an essential role in soil health, for they improve soil structure, water movement, nutrient cycling, and plant growth. Because of their function, healthy worms in large quantities are an indicator of healthy soil systems. In such soil, there should be an average of 15 to 100 worms in a 30 by 30 centimeter area.
This is our research question. How does the quality of the topsoil in Bayview shipyard compare to the quality of the topsoil in Crissy Field? This is observed through the abundance of Lubricus terrestris, a Latin word I will never be able to pronounce, also known as the earthworm. Our hypothesis is the earthworm population in Crissy Field will be more abundant than the populations close to Bayview Hunters Point, due to the toxins residing in the topsoil and surrounding bodies of water.
Our independent variable is the location, and our dependent variable is the number of worms. Materials and methods. In order to perform the experiment, we needed mustard powder, water, shovel, a bin to hold worms, and sample vials for our soil testing. First off, we mixed approximately 2 teaspoons of water with 1 liter of 2 teaspoons of mustard powder with 1 liter of water. Then we poured approximately 1 liter of the mustard water over the testing site, making sure to evenly disperse the water throughout the area. We located the patch of soil and made sure to clear any top foliage with a shovel or rake to ensure water absorption. We waited at least 5 minutes to observe worm action. When a worm is spotted, we carefully removed from the soil and placed in the bin. Then we counted the total number of worms collected and then carefully put them back into the soil. To collect soil samples, we collected approximately 120 cc of soil in a pill vial. The vials were tested by R.J. Lee Laboratory.
So here's some of our data, which displays the increased populations of worms found in Crissy Field in comparison to the worms found in Bayview Hunters Point. Our p-value for this study of worm tests is 0.042. This illustrates a significant difference in data collection between the two sites. 0.042 illustrates that there is a 4.2% chance that our data was due to random chance. The 6 worms that you do see from Bayview in both graphs are from remediated soil.
This is a map displaying the average word count found in our locations. And this is a map that shows where the worm tests were performed.
By the worm tests, we also collected soil samples. Here's our p-value. Again, there is a 4.2% chance that this data was all due to random chance, which is good because it proves our research is significant.
This data displays our soil sample averages of chromium, lead, and mercury. As you can observe in Bayview, there is a much higher amount of toxins tested in parts per million. The remediated soil in the areas do show a smaller amount of toxin shown, and Crissy Field shows the healthiest soil.
As you can see, these are our averages from 3 sites: the Marina, which is remediated soil, the Bayview remediated soil, and Bayview non-remediated soil. So Bayview non-remediated would be the soil from the shipyard. Here are our averages. As you can see and observe, chromium and lead are significantly higher on average in Bayview non-remediated soil than in Marina and Bayview remediated.
And this is a map that displays our data further. In yellow, you can see the average word count, and as in red, blue, and orange, you can see the 3 toxins we tested for. And as you can see, the larger the amount of toxins, the smaller the average worm count. And here is it a little more zoomed in.
And here's the Marina.
This is Bayview, and this is the Marina.
And so our discussion. So the main patterns that we see in our data is an increased number of worms in Crissy Field. This is true to our hypothesis. The results demonstrated a healthy soil quality in Crissy Field and unhealthy soil in Bayview. This is important to address that the only worms that we found were in remediated soil in Bayview. Additionally, these worms were very thin, signifiers of poor health. The worms in Crissy had a large circumference and were more vibrant in color. Those are the worms below. The worms at the top are from Bayview. We did not find any worms in original Bayview soil. This aligns with the current science proving the toxicity of the Bayview Hunters Point area and our soil samples. The fact that we found more worms in Crissy is very telling of the neglect that Bayview has faced.
We tested an increased amount of heavy metals in Bayview Hunters Point, specifically mercury, lead, and chromium. This data proved the toxicity of Bayview Hunters Point. and aligns with our worm collection data. The long history of land abuse and testing explains the lasting impact of the heavy metals on the land. Though the remediated soil tests for less heavy metals than Bayview Hunters Point soil samples proper, one questions how long a waterproof plastic seal with 3 feet of soil on top will hold out from these dangerous chemicals seeping through. The remediated soil is only present in the newer, expensive developments that are actually further gentrifying the community, offering no solution for the Bayview Hunters Point community itself. These soil samples were taken from both the shipyard proper and more residential areas, explicitly displaying the toxins in the Bayview Hunters Point community, the toxins the community is exposed to in their everyday life. And this correlation between lower levels of lead, mercury, and chromium and higher worm count average in Crissy Field, and then when there's higher levels of lead, mercury, and chromium, there is a lower worm count average. And this graph displays that information. As you can see, the red demonstrates the worm count, and and the blue is the heavy metal, and as there are less heavy metal tests, there are more worms.
Finally, this is Marie Harrison. She dedicated much of her life to protesting the contamination and development of Hunters Point Shipyard. She lived in Bayview for 60 years and worked at the shipyard when she was 18. She died last year of chronic lung disease which she believed she had due to shipyard pollution. She is just one of very many. The abundance of worms found in topsoil correlates to the sense of urgency and obligation a given government agency feels to remediate that soil. It turns out that while our research concluded that higher levels of heavy metals correlate to a decreased amount of worm population, it also turns out that the amount of worms in your backyard turns out to be a signifier of privilege. Thank you.
And a special thank you to Spencer Houston and Roger Wu, who were essential roles in this project.
Perfect. Nice work, both of you. That was a great presentation. And now, if any of you watching at home have any questions for Caroline and Grace about their project, please leave them in the comments section. And then I guess kind of just to kick things off for myself, I would love to hear you both share kind of why you ended up picking this topic, or kind of what jumped out to you about kind of these areas in your local neighborhoods and region.
Yeah, I can go first. So first of all, this is a project I'm researching Bayview Hunters Point for a service project in school. And I was really intrigued when I came across the Tetra Tech scandal. And I realized that I live next to Crissy Field, where it used to be a military and airfield base. But then on the other side of the city, we see Bayview. And while it also used to be a Navy base owned by the government, it has not been remediated. So investigating the variance in remediation was very interesting to me because it also correlates to discovering more about my own privilege in my own backyard.
And when Caroline and I were brainstorming project opportunities, we both decided that it was important to us that the research we would be doing would have a larger impact and demonstrate something bigger. And so we came across this because it intersects kind of the social impact and service aspect that we both value and the scientific studies.
Perfect. Well, that's great. It's always exciting when people get to kind of do things they're both interested in scientifically, but then also have a bigger, broader meaning. So as you went out and collected your data and saw your findings, were any kind of unexpected things that you found, or what were some of the highlights of the actual physical data collection yourself when you were doing that?
Yes, I don't know if any of you have seen the movie Erin Brockovich, but I felt like Erin Brockovich doing these soil experiments. Taking samples in the shipyard felt like a mission, and it was very exciting. Grace can attest to that. Also, just finding higher levels of non-heavy metals, but finding high levels of arsenic in the Crissy Field, that wasn't included in our presentation because it is not heavy metal, but that was very intriguing to us, and that can be included in further research and further investigation.
And I think getting our soil samples back with all this data about heavy metal testing and to be able to quantify what was going on in these places and see it in numbers, I think that was just like looking at the data and then transferring it into these like visible graphs that you can really see this difference. Like that was just so surprising and shocking, yet in some ways also expected.
Cool. So one last question for you here. So basically, if you could add any other factors to your experiment, kind of what might you add, and kind of to do this in the future?
While we can test for worms, we can't really test for humans. That's not in our capability yet, but that would be really intriguing to us, especially because of all the health effects on worms as an organic organism and material. We are also humans, organic. So by testing humans, we would probably see the same negative health side effects. And so that would be interesting to test for.
It would additionally be intriguing to look at the historical kind of biome of the area and see what previously plant flora and fauna were found before it became a shipyard, and compare it to today's tests and today's kind of the visible like flora and fauna in the area.
Perfect. Well, those are two great questions, and thank you both for sharing this awesome presentation. I'm really excited to see it, and then especially all the data from the heavy metals, and how it kind of matched with your earthworm findings. It's kind of a really neatly packaged experiment. So big virtual round of applause for Caroline and Grace and their awesome presentation. So thank you both. And then up next, we have Kaya, who's going to be joining us, who is going to share her presentation. How's it going, Kaya? And I will pull up your presentation here, and let you go to it. I think you're muted, but I think you're ready to go.
Hi everyone, my name is Kaya, and I wanted to learn more about how moisture content of soil affected the pH level and nutrient levels of the soil. So, like I said, I wanted to find out how moisture levels affected the pH level, which is how acidic soil is, and the nutrient content of the soil. So, the nutrient levels I chose to research were phosphorus, nitrogen, and potassium, which are all essential for plant growth and plant development. And I took my data from Prosser and Boca Lake, which you can see there, along with and as you can see in the maps, just to give you a visual, right there is Lake Tahoe, and then right above are the lakes that I took data from. And the why of my experiment, so I kind of wanted to research something relating to climate change, and I wanted to see how the moisture level affected the pH level, or if they did, and how they affected plant growth. And the pH level is relating to carbon dioxide acidification, which is why it's related to climate change. And just for some background information, acidification is when carbon dioxide is dissolved into water. So, and that relates to my question or my research topic, because I wanted to know if the soil was more wet, if there would be more carbon dioxide being dissolved into the water. And I did this experiment using a moisture probe to test different moisture levels, and to keep it as consistent as I could, I tried to take similar moisture levels, so around moisture level 4 or 5, which is pretty dry, 7 or 8, which is around mid-level moisture, and 9 or 10, which is pretty moist. And then I used a soil testing kit to find pH level and the different nutrient levels. So you can see the picture on the right there shows the materials that I used, and I collected this data on November 1st. So my question is, how does the moisture level of soil affect the pH level and nutrient levels of soil? So my independent variable was the moisture level, which I kept consistent through the three different moisture levels for the different places I took, and I wanted to see how it affected pH level, phosphorus level, potassium level, and nitrogen level. And I thought that the soil with a higher moisture content would have a lower pH, meaning it's more basic and less acidic. And I think that it would have higher nutrient levels because of living things need water to survive. And I think that there would be more living things with the water and more nutrients in the soil. So here's some of the materials I used. So on the left, there's the moisture reading probe. And then I used the soil reading test, which had those little containers in with tablets that I used to determine the nutrient levels, and plastic cups. And I also put a tape measure in because I did measure how far away each of the samples I took were, but it doesn't matter as much because I just measured moisture level over how far away it was. And what I did, so I first took the moisture levels, as I mentioned, from a wet, mid, and dry areas using the moisture probe. And I tried for the wet, I tried to get as close as I could to the water without actually being in the water to try to get very moist soil. And then again, very far away from try to get dry soil between. And at the bottom, you can see the different tests I took with the 6 types of soils I took, 2 different places with 3 different moisture levels in each of those places. So this is just a quick graph to show you the dry was again level 4, mid was around 6 or 7, and the wet was around 9 or 10 from the different samples I took. And then this is a graph which shows you the pattern of the moisture level to pH level. So in this graph, you can see like that light blue line in the middle kind of shows you that as the moisture level increases, so does the pH level. So it becomes more basic and actually less acidic. However, as you can see, it says that our squared level is 0.234, which tells you that the moisture level has some impact on the pH level, but it doesn't mean the moisture level causes pH level. So if that number was 0.5 or above, that would mean that there would be a significant impact. However, since it's a lot lower, it means that the moisture level doesn't actually have that big of an impact on the pH level. And just for some background information, the average pH level in Truckee is about 7.5. And from the data I found, it was around 7 to 7.5. And the neutral soil level, like the not too basic, not too acidic, is around 6.5 to 7.5. And then lastly, this graph shows you moisture level to the different nutrient levels. So you can see that while there is some difference between the nutrient levels in the dry, mid, and wet areas, they're all very, very low. So what I can conclude from the data I found is that from the pH level, as the moisture went up, so did the pH level. So meaning as it was more wet, it became more basic and less acidic. And also, as I said, the moisture level didn't have a huge role with relating to pH. And the nutrient levels, so as I said, they're very low, which makes sense for the observations I made about the locations. There was very, very little plant growth, which makes sense because plants need nutrients to survive and grow. Also, just to note, where I live and where I took data, there are a ton of pine trees, and pine needles tend to be super acidic, which is not great for plant growth. So that could have been one of the factors of why there wasn't a lot of plant growth where I took my data. So some things that I could have done to make my research a little bit better, because my results were actually the opposite of my hypothesis. I could have kept more constants, but I also didn't count for a ton of outside variables. So human impact, I didn't really account for that, or plant impact, which I later learned that, as I said, pine needles are super acidic. I didn't account for temperature or textures of soil. So I took soils from different places with different moisture levels, which some places were had more silt quality, and others were more rocky. For the future research that I could do, that I would want to test in relating in relation to my project. So I would want to maybe test different pH levels of the same soil. So I would maybe take the same soil, but I would instead add water to it instead of testing different moisture levels, and then see what the pH level is. So I have more constants and less outside variables. And then also, I think it'd be interesting to take the same plants and put them in different pH levels of soil, and just see how well the plants do and see what plants thrive best in different types of acidic or more basic soil. And then the big picture. So why does this matter? So as carbon dioxide gets released into the air, this leads to more carbon dioxide seeping into the soil and to the water, which then leads the soil to be more acidic, which, as I have mentioned, is not great for plant growth and plant development. So right now, climate change is a huge issue because more carbon dioxide is being emitted into the air. And as you know, we need oxygen to live, which is what plants produce. So to be the CO2 being emitted into the air, and it's important to research this because plants provide oxygen for us to live. So yeah, that's kind of the bigger picture of why I wanted to research acidification and pH level of soil. So yeah, questions, and here's my works cited.
Awesome job, Kaya. Nice work. So if any of you watching at home have any questions for Kaya, just pop them in the comment section and we'll do our best to answer all of them. One question that I had were there any challenging or difficult parts in terms of the data collection when you were going out and collecting these sediment samplers, soil samples in these different areas?
Yeah, one of the challenges that I faced was finding the same moisture level in my soil. So I wanted, since I would kind of had to find my independent variable, the moisture level, it took a couple of tries to find the same moisture level. So like the first place I went to, I kind of decided like, this is what I'm going to do. And then going to the second lake, it was kind of difficult to find the same moisture content in the soil. So that's just one of the challenges that I faced. Yeah.
Perfect. And so one thing you talked a little bit about in your presentation about kind of maybe the time of year that you did this experiment. Do you have any thoughts or just guesses as to what it might look like if you did this experiment in a different time of year or a different season as opposed to here in October?
Yeah, so the lakes that I took data from, they actually drain the lakes during the winter. And so I think that maybe the data might have been different when the lake levels were higher because maybe there would be more nutrient levels, more living things. So I think maybe my nutrient levels might have changed and that might have impacted my pH levels. So I think that the time of year definitely could have impacted my results.
Cool. Yeah, it's awesome. Sometimes us as scientists, we want to just collect data all the time, and we don't always have that information. But yeah, so awesome job, Kaya. I really appreciate you sharing this awesome stuff. I kind of live near some of these reservoirs, and so it's always neat to hear about what's going on there under the soil. And so thank you so much. We'll give you a big virtual round of applause for all of us watching here in the backstage. And thank you so much. And up next, we have our next student. We have Liam, who's going to share his presentation with us. Let me just pull that up here. Perfect. And welcome, Liam. Perfect. I think Liam is joining us just via audio. But I think you're all set to go. Let me pull this banner out of your way. And Liam has an awesome presentation. I'm really excited to share with him to share with all of you about wildfire prevention, kind of one of the most unique projects that we have today. So Liam, with that, I think you are all ready to go, and I'll get myself out of your way.
All right. Hello, I'm Liam, and my project was on wildfire prevention. Wildfires are an important subject of study mostly because of the destruction they cause. Over 4 million acres of land have been decimated by wildfires this year in just California, and there have been a confirmed almost 10,500 structures burnt out because of wildfires. A lot of the information that I looked at and used here are regarding one specific method of wildfire severity reduction, which is simply removing excess flammable material through mechanical means, wood chipper, to reduce the amount of material burned by a possible blaze. And a very important note, I did not cause any fires. I didn't personally collect the information displayed here. The testing and results were carried out and recorded by the Missoula Fire Lab team. However, I did all of the analysis that is displayed here. The research question I had was whether wildfire production techniques would help reduce the severity of wildfires without damaging the environment too much. And I believed that wildfire prevention techniques would reduce the severity of wildfires without damaging the environment, simply because that's kind of their purpose, to reduce the damage wildfires cause. The materials used by the Missoula Fire Lab team were a bunch of scientifically planted pine stands and a wood chipper, as well as fire. The methods the scientists used were they took the forest plots and divided them into segments. Half of these plots were treated with half of these plots had their excess material chipped up into coarse pieces, and the other half were chipped up into more fine pieces, closer to dust than wood shavings. Or tan bark, I guess. 1 year later, they burned half of the plots of each type and measured the effect the fire had, from how fast it spread, to how long the flames were, and how burnt out the soil got. Then they came back in year 2 and burned the rest of the plots in each group and recorded the results. This is a graph of the flame length recorded between the tests on year 1 and year 2, with coarse or more tan bark like chippings outlined in blue, and fine, more dust like chippings outlined in red. The p-value of the p-values of year 1, or the p-value of year 1 was 0.872, which was a very minuscule difference between the two. But in year 2, it was much, much smaller, only 0.0043, which means that in year 2, there was a very significant decrease in the length of the flames when tested on coarse chippings rather than fine chippings. That's important because if a flame burns lower and it doesn't have quite as long flame lengths, then it's less likely to be able to burn up to the more flammable leaves on the roof, in the canopies of trees. To discuss the results, the coarse treatment reduced flame length during burning around 2 years after the original treatment of the plots. Larger wood chippings reduced flame lengths by likely reduced the flame length by slowing the regrowth of smaller plant life in the area that was treated, where smaller, more dust-like chipping only served as fertilizer for future plants. This information is useful to forest managers and people that work to prevent wildfires because it shows evidence that a way of reducing the destruction caused by wildfires is effective in preventing larger blazes due to the destruction of the canopies of trees. The tests in this the tests in the data primarily only involve trees of one specific type, pines at a specific age of around 30 years old in a specific climate, which means that if this test was recreated in another place with other trees, perhaps of different ages, you would have likely different results. If I had the capability to do more research in the future, I would likely test with more variables, like different types or ages of trees, different sized chipping treatments, perhaps much larger chips, or something in between the two sizes of chippings in the tests that were previously recorded, or longer or shorter waits between iterations of testing. These are the works that I drew upon for my information and to conduct my analysis, and that's all. Thank you.
Well, nice work, Liam. Thank you so much for sharing this awesome question. If any of you watching along have any questions for Liam or any of the students, feel free to put them in the comment section as we go along. We love to hear your questions and go for that. And just to maybe start things off, Liam, I just wanted to ask you why fire? Why did you choose to investigate fire in your research project?
I'm from California, so wildfires have been kind of a pretty important part of everything that's been going on here for the last few months.
Nice. And so with that, I know you had some restrictions in being able to not being able to kind of start your own fires and kind of collect those data. If you had any thoughts or suggestions on kind of this data taken from the Missoula Fire Lab kind of out in the Montana area, any suggestions or thoughts on kind of what you might do to more specifically learn or study about wildfire in the California area where we all live? Any suggestions or thoughts on what that could look like?
If there were supposed to be, or if there were going to be tests on the same method of reducing fire severity, you would likely, or tests would likely have to be done with much more varied ages of trees and likely more with much more diversity. Perhaps looking at the most commonly seen trees in San Jose and using those for testing.
Yeah.
Or using those types.
Yeah, that'd be really interesting to do, and I hope some scientists are able to do that before too long. The Missoula Fire Lab, for anyone who doesn't know, does some really cool stuff, and they have a neat website that you could look at through Liam's links here. Great. So with that, thank you so much, Liam. And up next, we have Carly, who's going to share her presentation. Let me just pull this up here. Great. And hi, Carly. How's it going?
Hello, my name is Carly Baker, and this is my presentation on the human impact of macroinvertebrates. Firstly, we should start off on what is a macroinvertebrate. It's not a commonly used term, but macroinvertebrates are small but visible invertebrates. Best known would be insects or arachnids. The ones I'm studying are aquatic macroinvertebrates, those that live in the lake or water. Here are a couple of examples of them on the macroinvertebrate index card. Keith? My hypothesis / question. My question is, are there more macroinvertebrates on the side of Boca Lake that is less impacted or more impacted? And my hypothesis is there will be more macroinvertebrates on the side of Boca with less human impact. Boca Lake, you probably already heard of it in Kaya's presentation, is a local lake up here that is widely used by tourists and locals alike. Why macroinvertebrates are important. Macroinvertebrates tell a lot about the lake and local nature's health, and humans we already know that we have a big effect on the environment, and by seeing how that affects macroinvertebrates, can see how that affects the lake as a whole. Methods. Firstly, I found 3 spots on both the impacted and the natural side of the lake. There were too many macroinvertebrates to count in the net that I found, so I would sweep the area 3 times, and then I would count from 1 square inch and then multiply it by the area to find an estimate of how many there I found. And then I would move on. I would repeat this for each spot on both sides. Here are my results. I found most of the macroinvertebrates in 1 point. I found them in point A, which was where I found the most, and point A, B, and C were all part of the natural part of the lake, while point Z I combined into the impacted part of the lake. You see, point Z didn't have enough macroinvertebrates in its entirety to be classed as 3 extra points. You can see here that the most were found in point A, with the second most being point B. I found nothing in point C and 0.666 repeating in point Z. And the amount of macroinvertebrates per day doesn't change that much, and that helped me know that this was a consistent amount of data. This was a consistent difference in points. And here's another graph that showed the difference in each day in the natural and impacted. You can see how little the human impacted had macroinvertebrates, while the natural had much more, ranging in the 200s mostly. Discussion. I found that my hypothesis was correct. There are many more macroinvertebrates on the side with less human impact. And I learned that from one interesting spot that macroinvertebrates are very localized and that they vary from spot to spot. I found most macroinvertebrates were at point A, as I mentioned before. Why I think this might be the case is on one side of the lake, the impacted side is where most tourists are, most locals also. So they have more cars, and therefore it's flatter, and there are more rocks at the shore because everything's more smoothed out. While the other side is very sloped, and no one goes over there, and so there's less activity. And you can see both locations at the bottom. The left being the less impacted and the right being the more impacted. And if I were to do this experiment again, I would look at more locations, as I found they vary greatly, and I would do this during the height of activity, which is during the summer, as I did this during the fall. It's not something I could change when the amount of people was lessening. Thank you for watching.
Awesome job, Carly. Thank you so much for sharing that. So as people think of any questions that we might have for Carly about her projects, for some of those who maybe are less familiar with macroinvertebrates, can you maybe just describe a little bit, maybe like the types of macroinvertebrates you found, or some of the neat things that you saw in your data collection?
I found the majority of macroinvertebrates I found were small, almost clear little things, and the closest I could pinpoint them were to, I'll go back to the where the middle section, group 2, scuds. Though I couldn't tell exactly because, like I said, they were very small and there were many of them, but I found mostly them. I did find the occasional worm or larvae, but I found mostly just one type of macroinvertebrates.
Perfect. And then I also just maybe want to ask you, like, why did you pick macroinvertebrates, and why did you pick this specific lake to kind of ask your question about?
I picked macroinvertebrates because, as I before mentioned, that they are a great way of telling the health of a lake or any part of nature, basically, because they are very susceptible to pollutants and so on. And I chose this specific lake because it is it's local. I live near it, and it is also, I see many tourists there, and tourists and locals alike just there on the shore with their cars. And it kind of got me thinking about how this affected the lake's health. And so I thought this would be interesting to study.
Perfect. Awesome. Well, thank you so much, Carly, for sharing this awesome presentation. It was kind of neat to see the photos from where you were collecting the data and see what the different parts of the lake look like. And so with that, we're going to finish off with one more student presentation. We have Talia, who's going to close us out today, who's going to kind of take us back to the Bay Area and share a really neat presentation on kind of water quality and some of the things that affect it in there. And so with that, I'm going to add Talia and her presentation here. Hi, Talia. And I will get out of your way and let you go if you got that presentation button at the very top. Perfect.
Oh, this is. Okay, so hi, I'm Talia Rotman. I go to the College Preparatory School, a school in Oakland, and this is my project on urban materials and their effect on water quality. So to give you some background info on the big picture behind my research, farming and urban activity leads to contaminated runoff, and then this leads to a process called eutrophication, which I will go into more detail on in the next slide. So under farming, a big culprit for this contaminated runoff would be excess fertilizer. And then under urban activity, there are multiple different reasons. One of them is that with less plant growth, the soil is less healthy and more erodible, and so more likely to end up in runoff. And then also the wear and tear of driving and human activity pulverizes asphalt and concrete in the land below it, and then leads to more sediment that ends up in runoff. So then this runoff ends up in rivers, lakes, and oceans, and some of the materials can directly lead to a toxic environment that is bad for aquatic animals, and it also indirectly can lead to a toxic environment through eutrophication. So what is eutrophication? Eutrophication is when excess nutrients lead to increased algae growth, and this is because the nutrients are made to promote plant growth. And then as the algae is growing, it uses up these nutrients to the point where it's used up all the nutrients, and then the algae suddenly dies. So then at this point, the algae gathers at the bottom of the body of water and decomposes. And when it's decomposing, this is through the process of cellular respiration. So cellular respiration takes O2 out of the water and adds CO2. So then with less oxygen, this suffocates the fish, leading to massive fish kills. And then another issue with this process of eutrophication, during the increased algae growth part, there can be toxic algae blooms, which are very harmful to humans and pets and other animals. So then some background info on the nutrients. Let's start with phosphate. Phosphate is in fertilizer, manure, industrial effluent, and sediment. Phosphate is very essential to algae growth, so the more phosphate, the more algae. Contributor to the often algae blooms in a local lake here in Oakland called Lake Temescal is this excess amount of phosphate. Some background info on ammonia and nitrate. They are also in fertilizers and other forms of organic waste. And in high levels, unlike the other nutrients, ammonia on its own can be directly toxic to aquatic life. So in this way, algae can actually initially be helpful by decreasing the ammonia concentration while it's growing. However, when algae decomposes, ammonia is released back into the body of water. So a healthy concentration of ammonia is 0 parts per million, and it starts to become very dangerous for fish at 1 part per million and above. And then lastly, some background info on pH. pH is the measurement of H+ ions, and the more H+ ions, the more acidic. The less H+ ions and more OH- ions, the more basic. So when alive, algae increases the pH, making it more basic because it is photosynthesizing. So it's taking CO2 out of the environment, and CO2 is an often culprit for acidification. However, when decomposing, algae does the opposite. and decreases the pH because it's adding CO2 through cellular respiration into the environment. So for most freshwater aquatic life, pH should be close to neutral at around 7, maybe a little below, maybe a little above. And while aquatic life can tolerate some shift in pH, sudden and big shifts are harder to handle and can lead to the death of many aquatic animals. So to give you an example of this process of eutrophication, as mentioned earlier, there's a lake here in Oakland called Lake Temescal. And in the first image, you can see this is the pipe where the water first enters the creek that leads straight to Temescal. So I measured the phosphate concentration at this point, and it was already at 1 part per million. And keep in mind, the levels as low as 0.1 parts per million have been linked to algae bloom. So this is already way above the recommended amount. And then in the second image, you can see this map of Lake Temescal, and it gives you an idea of how urban this lake is. And in addition, right now the surface looks pretty clear in this picture, although it does have a green hint. There isn't really too much surface algae or other plants such as duckweed. However, in this last picture right here, that's a picture I took about a week ago at Lake Temescal. There is this thick surface layer of green algae and duckweed that is covering the entire lake. And it has been like that for a while now. And this issue of eutrophication has actually gotten so bad at Lake Temescal that they have installed these nano oxygen bubbler tanks that inject really small bubbles of oxygen into the lake to try to combat this process and create some oxygen to sustain the fish population. So then this brings you to my research question. My research question is, how do different urban materials affect water quality? So the independent variable was material, and I chose asphalt, concrete, soil, and fertilizer. And then I was asking, how do these materials affect the dependent variable of water quality? I quantified water quality as pH and ammonia, nitrate, and phosphate concentrations. So my hypothesis was that the fertilizer will lead to water with the most nutrients because they are made to promote plant growth. So therefore, they will lead to the most algae growth. And my materials, to give you a little bit more detail about the materials I was testing, I tested a Kellogg's fruit and vegetable fertilizer, and I gathered some asphalt from the side of the road. In the first image here, you can see the site where I collected it. And keep in mind, this is about 20 feet uphill from this storm drain in the second image. So this is material that will very likely end up going down that storm drain and into a body of water the next time it rained. And then my third material was concrete. This is just concrete from my backyard. And I ended up hammering a big chunk just so I had some smaller bits to work with for my testing. And then I also tested some dirt from a dry grass field in my backyard. And keep in mind, this isn't the healthiest of soil. There hasn't really been that much plant growth in it. So it's super erodible and dusty and thin, yeah. And then in addition to comparing these materials to each other, I wanted to add sort of a control. So I created some samples with just tap water as sort of a baseline. And then in addition to those, I used 20 clear identical plastic cups, measuring cup, a scale, a sieve, and then for the testing materials, I used the API Pond Master test kit. And in that kit, there were test tubes, pipettes, a pH test solution, phosphate test solutions, ammonia test solutions, a nitrate test solution, and lastly, a color chart to see what the resulting colors meant in terms of concentrations. So then that takes you to my method. I began by sifting the materials, as shown in the first image. I wanted to, for the most part, reduce surface area of the particles as a factor that could contribute to how much nutrient the material leaches. And after that, I placed 0.3 ounces of each material in a plastic cup. I had 4 replicates of each material and also 4 replicates of the control, which was just water. And then in the third image from the top, you can see how I randomized the cup layout. This was to control for factors such as time and light exposure. And then I added 100 milliliters of water to each cup. I mixed each cup for 20 seconds, and then I let it sit for 24 hours. So the next day, I came back and tested the phosphate, nitrate, ammonia concentrations and the pH. So then for my conclusions, for the average phosphate concentration graph measured in parts per million, I concluded that the fertilizer led to water with significantly more phosphate than the other materials. As seen in the graph, the fertilizer is at 8 parts per million more than the second place of soil. And then my other conclusion was that other materials, with the exception of concrete, increase the phosphate concentration as well when compared to tap water. So my statistical backing for these conclusions is that the main p-value of 0 very strongly indicates that these patterns are true. Secondly, the F statistic of 937 indicates that the averages are reliably different. However, the Tukey test indicates that asphalt compared to concrete, asphalt compared to concrete, asphalt compared to soil, and concrete compared to water were not significantly different when compared to each other. So then my conclusions for the average nitrate concentration graph, also measured in parts per million. The soil increased the nitrate concentration the most. This is illustrated clearly in the graph. And secondly, the asphalt and water did not have a measurable concentration of nitrate. So the statistical backing for these two conclusions, the main p-value of 0 very strongly indicates that these patterns are true. Secondly, the F statistic indicates that the averages are reliably different. And lastly, the Tukey test indicates that all combinations are reliable. And then a little disclaimer about this graph, the reason why you don't see fertilizer displayed is because the fertilizer had started off with a slight orange tint, and especially for this color assay, it really disrupted the results and it was completely illegible. As shown in that little picture, the only options were arranged from blue to purple and ended up as like a dark yellowy green brown color. And then for my average ammonia concentration graph, also measured in parts per million, I concluded that concrete and soil led to water with the most ammonia, with soil leading to the most. And then secondly, fertilizer and asphalt do not increase the ammonia concentration. So my statistical backing for these conclusions is that the main p-value of 5.475 times 10 to the negative 13th strongly suggests that these patterns are true, being way smaller than 0.05. And secondly, the F statistic of 211 strongly suggests that the averages are different. However, the Tukey test indicates that I cannot reliably compare asphalt to water. And then lastly, my conclusions from the average pH graph. The concrete increases the pH the most when compared to water, whereas soil decreases the pH the most when compared to water. So my statistical backing for this is that the main p-value of 2.504 times 10 to the negative 8th strongly indicates that the overall patterns are true. Additionally, the F statistic of 47.25 strongly indicates that the averages are not equal. However, the Tukey test indicates that I cannot reliably compare asphalt to soil, asphalt to water, and fertilizer to water. So what does this all mean? That brings you to the discussion. Going back to my hypothesis, fertilizers will lead to water with the most nutrients because they are made to promote plant growth. I would say that this data backs this up. As seen from the graphs, the natural materials led to more nutrients. Soil led to the most nitrate and ammonia, and fertilizer led to the most phosphate. I would say this is predictable since these organic materials were derived from the decomposition of life, so it makes sense for them to have the most nutrients. Additionally, the natural materials led to lower pHs. This is also understandable because high nitrate levels, nitrate often being in fertilizer and soil, often leads to lower pH. And then lastly, concrete increased the pH. This can be explained because a common ingredient, lime, in concrete, which contains calcium carbonate, can increase pH as CO3 2- binds to hydrogen ions. And then some sources of error. One being that the mixtures were not homogeneous. An example of this was when I collected the asphalt, there were other bits of debris in there, such as like little bits of maybe ground up leaves that I wasn't able to separate. So this may have impacted the results of how many nutrients were leached from it. And the same goes for the other materials. Secondly, the fertilizer-soaked water slight orange tint made the color assays a little less accurate, even for the ones where I could try to make out what the color was. And then for the third reason, testing took a while. Some samples may have had longer/shorter soaking time, even with the randomized order I created to try to limit this. And then lastly, even though sifting gets rid of surface area as a factor to a certain point, even after taking out all of the pieces above a certain size, there could still be a variation in exactly how fine the materials were. And so this experiment has led to some more questions, like how would algae growth differ when placed in these samples? And then how would this range of algae growth in turn affect the water quality? And then I also want to ask what materials erode the most. So this will help answer the quantity of each of these materials that would enter a body of water. And then I'd also want to ask, why does Temescal in particular have such an influx of nutrients? And then lastly, how can fertilizer runoff be reduced while still allowing for abundant agriculture? And then most importantly, what can we do? So, well, most of us don't own large farms, so we can't reduce agricultural runoff on the large scale. As in an urban environment, there's still a lot we can do if we have a garden in our backyard. So to reduce nutrient pollution, we can be really conservative with our fertilizer use and don't overuse it. So to remedy this, you can plant native plants. Native plants are more adapted to the natural environment, so they need less fertilizer, if any at all. And then secondly, don't apply fertilizer before windy or rainy days. Wind and rain can take this extra fertilizer and take it away from your plants into the street, possibly down storm drains and into a body of water. And for very similar reasons, don't overwater. The excess water can again lead to fertilizer ending up where it shouldn't end up. And if you want a more extensive list of what you can do as a gardener, go to the EPA website under nutrient pollution. And here is my works cited for the information and images. And I also want to thank Spencer Houston for teaching this class. I learned a lot about the scientific method and how much creativity it takes, which I kind of underestimated. And also for being super helpful with my questions about specifically eutrophication and water pollution. Thank you for listening, and let me know if you have any questions.
Awesome job, Talia, and thank you for that. It was super fun to work with you and all the other students in this program. As you kind of said, the creativity that each one of the students brought, it's why we had so many different presentations, was awesome. And so I just wanted to start us off with one question here. Of, you kind of tested these materials in your presentation. If there are any others that you would add, if you had kind of infinite time and resources, what might be other materials that you might want to look at or would be interesting to look at, kind of related to your question?
Yeah, I don't know about any different materials, but I would definitely want to change the location. I think taking materials from my backyard was a bit removed from the issue of Temescal. So maybe I'd want to take locations upstream from Temescal in areas where I know water is being taken down into Temescal from that location to see what might be affecting the water quality there.
Oh, cool. And then my last question for you, as we kind of wrap things up, is like here in California, we've kind of had a really dry summer, and then it's just in the last couple of weeks started to rain. If you had any thoughts on how kind of precipitation in this new weather change that we've had might impact some of the area that you studied and what results that could have.
Yeah, I was thinking about that recently because it has been the first time it rained in a really long time. And I think it would have been really interesting to measure things like phosphate concentration before the rain and then measure it after, because I'm sure there's all this sediment that's just sitting around for a long time that's being run into Lake Temescal.
Perfect. Awesome. Well, thank you so much for sharing your project. I'm going to pull you out of the stream and then just finish off with a couple notes. So I wanted to thank everyone who watched today. This is a testament to all the awesome creativity these students brought to this program. And they not only had to learn how to think creatively and problem solve all the things that may have come up during their data collection that didn't make it into their presentations, but these science skills are things that they'll take with them forever. And so with that, if you have anyone that you know who might be interested in doing a project like this, the next opportunity to do so with Headwaters is during our virtual research program that is starting February and running to April. It'll be just like this, but instead of these end presentations, students will write a scientific manuscript and kind of mix things up at different points of the year. But we also have similar research programs coming this spring and next summer as well. All of the information on that is on linked through our Facebook page or on our website, headwaterscienceinstitute.org. And thank you all so much for joining and watching these presentations today.